Hello, everyone. I think it's time to get started. Good morning, good afternoon, good evening, and for some of you, good night, I think. And welcome to our webinar series, New Tools of the Trade in Life Sciences, Technology that Enables Breakthroughs. And today's webinar topic is 3D printing personalized tissue models. My name is Renate Walzbauer. I'm the lead editor of this program here at BioProtocol, an open access online journal whose mission is to make the research more transparent and reproducible. Before we begin today's webinar, I would like to make a few quick announcements. If you have a question, please feel free to post them in the Q&A section or the chat sections throughout the talk, and Stephanie will address them at the end. In addition, the webinar will be recorded and posted later on online. So if you want to revisit it, you can do so. Okay. It is my distinct pleasure to introduce the moderator of today's webinar, Milena Restan-Perez, who graduated with a Bachelor of Engineering in Biomedical Engineering from the University of Victoria in Canada. Her research work focuses on engineering neural tissues using 3D printing, fibrin-based bioinks, and stem cells to study Parkinson's disease as well as glioblastoma multiforme, for which she is currently also testing a potential new drug delivery system. She is the first author and co-author of several papers on 3D bioprinting, including an extended protocol describing how to bioprint mesenchymal stem cells using 2 different bioprinters, which was published here at BioProtocol. And with that, Milena, thank you so much for being here. And the floor is all yours.
Perfect. Thank you so much. Thank you for that introduction. I'm very happy to be here, and I'm very happy to be introducing my boss, the best boss. She didn't pay me to say that, and someone who I've considered a mentor throughout my biomedical engineering career. Dr. Stephanie Willard is a full professor in biomedical engineering at the University of Victoria, where she has dual appointments in the Department of Mechanical Engineering and the Division of Medical Sciences. She also holds an appointment with the School of Biomedical Engineering at the University of British Columbia, and she recently founded the startup company Axolotl Biosciences that sells high-quality bioinks for bioprinting human tissue models. She's an active member of the steering committee of the BC Regenerative Medicine Initiative at the Stem Cell Network. She also serves as a staff scientist at the Creative Destruction Lab, and she served as the acting director of the Center for Biomedical Research and the Biomedical Engineering Undergraduate Program at the University of Victoria. She also served as the president of the Canadian Biomaterials Society. And if all of that wasn't enough, she was also elected to the Royal Society of Canada's College of New Scholars, and Engineers and Geoscientists of BC awarded their Teaching Award of Excellence in 2021. And with all of that, all those titles, I'll pass it on to Stephanie.
Awesome. Thank you so much. That makes me feel like I need to take a nap. And I think if it's all okay, I will start my talk. And yeah, I'm really excited. Thanks again for the opportunity to be here. I'm really excited to talk about some of our work, and Milena actually did a lot of the work I'll be talking about today. And as she mentioned, we are located in Victoria, British Columbia. So we are on an island, as you can see here. So our campus is inside of a ring road. And if you ever would like to come visit us, you can come check out all of our different bioprinters. I think we have 6 different bioprinters. For those of you who are in Canada, the Stem Cell Network does offer technology transfer awards. So if you do want to come out and learn about bioprinting, we'd be happy to host you and help you support your applications for that. And so today I'll be talking about some of the work that we've published and really looking at how we can use 3D printing as a way to generate these personalized tissue models. So since I'm in my home office, let's see if this will work with the BioProtocols background. So I think a lot of people are familiar with traditional 3D printing, which this is an axolotl I'm holding up that's 3D printed. So you have a CAD file, a design file that contains specifications, and then with traditional 3D printing, you heat up a plastic filament that gets then secreted into forming the desired shape from this file. And usually with the plastic 3D printing, you end up with a hard object. 3D bioprinting is a similar process, but instead of using filaments that you heat up, because cells don't really like to be at extreme temperatures or conditions, usually you use specially designed bioinks when you're generating these 3D printed structures from your tissue design file. And then once you have your tissues, you can culture them and use them for things like drug testing or studying fundamental biology, as well as therapeutic tissues, which one of our collaborators, Aspect Biosystems, just recently announced a $2.5 billion deal with Novo Nordisk to sort of use their printers to make therapeutic tissues for our applications like that. Also, 3D Biotherapeutics, who spun out of Cornell, they actually have put mesenchymal stem cell derived 3D bioprinted cartilage type tissues for mimicking, it's for people who have ear deformities, and they would scan the healthy ear, and then they've been using bioprinting to actually print and reconstruct the other ears and transplanting them into humans. So there have been examples of clinically implanted 3D bioprinted tissues, which means this is a really exciting area to work in, and there's a lot of really cool things you can do with these applications, especially as the field has been evolving. So why would you win a bioprint compared to some of the other methods? Well, back in the day, when I did my PhD, we would make our tissues by pipetting different mixtures of our protein hydrogel scaffolds into things and manually mixing them, which took a lot of time and effort and was not very high throughput. And so I remember back in 2015, when sort of the bioprinting companies were coming out with these technologies, I was thinking, oh, wow, this would be a really good way to automate these processes. And also because you're starting with a computer-aided design file, you do get better reproducibility compared to some of the other techniques, although we are still very interested in organoids, and we've made them in our lab before. The other thing that's really interesting is with bioprinting, you can potentially generate more complex structures where you have specific regions with different cell types inside of your tissue construct. And so you can really sort of get towards mimicking some of the structures you would find in an in vivo tissue as well. And although I don't really touch on it today, our group has published papers where we have actually bioprinted aggregates of cells, because I know there's a lot of people who work with organoids, and there are ways to actually print. And I think Cyfuse out of Japan, their whole method, their Kinsian method's based on printing spheres into tissues that then can be used as well. So you want to bioprint, what do you need to do? One is the method of printing. I'll talk about two different printers today. One's the Aspect Biosystems RX1, which is quite a high quality, can print tissue grade tissues on the relatively expensive side. Also the BioX from CellLink, which is more affordable, but and also in some cases easier to use, but can be a little bit more harsh on cells. So really there's like the whole gamut of different types of bioprinters. And you can also look around. There definitely been some open access papers published recently where people have actually been able to modify enders to be bioprinters if you don't have that much money to spend on an instrument, all the way up to things like the bioassembly bot, which actually can print in 6 axes and do all sorts of other really interesting technologies. The choice of cells, a lot of the stuff I'll be doing today, we actually print with around 1 million cells per mil to try and mimic the same density of cells that you would find in a human tissue. So number of cells, type of cells is very important when you're deciding what structure to make. Choice of bioinks, usually the things that are easiest to print aren't necessarily what's best for cells, and that's why we spend a lot of time working on making our tissue print product and optimizing that to get a printable version of fibrin. Tissue structures, our group is actually doing some work into machine learning to figure out what structures are printable and what aren't. But yeah, in general, like what shape and where do you want your cells to go inside of the structure is also an important consideration. And you can also incorporate growth and differentiation factors into these tissues as well, which also can help determine like how quickly the cells inside your tissues mature, can also affect their functionality. So obviously a lot of things to think about if you're going to bioprint. So one of the printers that we have used a lot in our lab, and we were, I think, the second group ever to get them, was is the Aspect Biosystems RX1. They use these microfluidic print heads that you can see here, which they call their lab on a printer microfluidic technology. And because you have different inputs, you can mix, well, in this case, they're just showing different colors, but you can have different types of cells. We often print with a crosslinker-based tissue printing. You can also flip between materials and cell types, so you can really print highly detailed structures using this technology. The other printer that I'll be showing today is the CellLink BioX, and there's also an X6, which sort of is similar technology, but the 6 refers to having 6 print heads. These are really interesting and versatile printers because they're really easy to use. And they're compatible with a number of different print heads. And I think you can actually use one of these things to actually do traditional 3D printing. So it's quite a versatile system, and they have a lot of really fun preloaded designs. I believe we printed like from their preloaded things an ear and a liver, and also we've printed some brains that actually looked like miniature brains with human cells as well. So there's lots of fun different shapes you can play with on here as well. And in terms of what we've been developing here, both in my research lab and translating out through Axolotl, is we essentially, and this was done by a team of sisters and my former PhD student, so Lila, Emily, and Laura de la Vega, along with in collaboration with the guys at Aspect, we came up with a printable version of fibrin that worked really well at keeping neural cells alive. So prior to this study, I think people who were bioprinting neural stem cells got about 50% survival, and here we were getting something like 90% survival. The other thing that was really exciting about this is that the structures we printed were very stable, and you could keep them in culture for 40 to 50 days, which again, at the time, most people who were printing neural stem cells were keeping their tissues for around 7 days. So being able to keep your tissues for a longer period of time is really important, especially for studying things like neurodegenerative diseases. And it also enables us to get relatively mature tissues in the case of our neural tissues. So you do actually see the electrical and chemical signals that you would see in mature tissues. And we just recently had a paper come out where we were able to do this with HIPSC, human-induced pluripotent stem cell-derived neural progenitors taken from patients with Alzheimer's. And you're also able to start to see some of the Alzheimer's phenotypes in them as well. And yeah, the original BioInk paper was featured in the 2019 Methods of the Year issue of ACS Biomaterials Science and Engineering. And then since then, we published a number of different papers talking about how to use the zinc, not just on the Aspect system, but as well as the Selling system. Yeah, and so I'll talk about some of the original paper that Milena had published along with Ruchi and Nadia. Ruchi is also currently a postdoc in my lab who's been doing my tax-funded postdoc with Axolotl Biosciences. And Nadia just recently stepped down from being my lab manager, but she also manages our nanotherapeutics cluster here at the University of Victoria. So this was a really interesting paper because essentially we, instead of using our traditional HIPSC-derived neural progenitors, in this case, the idea was actually to print with mesenchymal stem cells, which are much easier to obtain. You can get them from fat, you can find them in blood. And so the idea was to print them and then try to reprogram them back into neural tissues using a previously published protocol that had been able to reprogram cells in a 2D environment. And so here you can just see the setup. As I mentioned, you can see the Aspect print heads and then the BioInk and crosslinker along with the buffer solutions that are fed to the different things. And we'll actually get to watch this process in action in a little bit. In this case, you print onto a sort of a little piece of film on top of a vacuum chuck that takes away the excess liquid as you print. And then A and B are just showing the designs. We were printing little half domes as a way to sort of get at replicating a miniature brain structure. So as I mentioned, the idea here was to reprogram these MSCs into neural-like tissues. And so sort of the control groups were we were doing this in neural basal media with B27 supplement, LoopMax, and PinStrip. And then we actually added pure morphamine, which is a small molecule known to help induce neuronal differentiation, FGF8, fibroblast growth factor, LDN and SB, which are also small molecules. So this was kind of the reprogramming cocktail that had been previously shown to work in 2D. And you can also see the BDNF treatments. And the idea was that this actually would convert your mesenchymal stem cells and reprogram them into dopaminergic neurons, with the idea we could use these as a tool for studying Parkinson's. And so here's just some of the interesting data we saw. So in this case, normally you wouldn't see TUJ1 or tyrosine hydroxylase present in these mesenchymal stem cells, but when we treated them with these cocktails, we did see enhanced expression in these different groups. And you can see just comparatively, we saw highly upregulated expression of these neuronal markers. Tyrosine hydroxylase in particular is expressed by dopaminergic neurons, so it's quite exciting to see that in our cultures here. And then Milena went ahead and quantified these. And so you can see huge increases in the amount of the cells expressing these markers. The 2D protocols I mentioned had already been published. So that was kind of interesting to see the differences there. But we were getting relatively high rates of conversion inside of our tissues. And this is exciting for a number of reasons. One, as I said, this is actually a little bit of a simpler path to getting a dopaminergic neural tissue for drug screening. And also a little bit more accessible for trying to make these patient specifics. For those of you who've worked with making human-induced pluripotent stem cells, normally you're taking either a skin punch or a blood draw, and then that's what you're reprogramming. And there's often a lot of issues doing the quality control and getting your desired differentiation once you've made your HIPSC lines. And so it's a little bit more direct to just directly reprogram a mesenchymal stem cell, which are much more accessible from patients into these types of neural tissues. Although we don't really get into it in the paper, the other thing that I think is something that's quite interesting, because this is, I think, one of the first examples where we had combined bioprinting with reprogramming, is to think about this not just as a tool to make tissues, but as a way to study how reprogramming can work. And so there's been interest, for example, our group had published a paper where we had used a small molecule cocktail to convert brain cancer glioblastoma cells into neurons. And then when we tried to do it in 3D, it didn't work as well. And so I think this type of system is a good way to study, like, can you achieve programming in a 3D environment before moving on your treatment to something like an animal study. And so because of the excitement around our work in this, we were then asked to write this up as a protocol. And again, you can see Melinda, Nadia, and John, who's been amazing. He's worked for me as a lab tech over the past year after working for me as an undergrad, and he's leaving us to go to Vancouver. So if anybody in Vancouver is looking for an amazing scientist to hire, John is who I would highly, highly recommend. And so, yeah, we decided to turn this into a very detailed protocol. One of the things, and probably why a lot of you are here listening, is one of the things that makes 3D bioprinting so cool is it's so inherently interdisciplinary. But a lot of the people who've made the bioprinters, and I'll say this is the same for our friends at Aspect, it's spun out of an electrical engineering department. It's a very mechatronics project, moving print heads, controlling flow rates of bioink, things like that are that come from a very mechanical, mechatronic type perspective. And so then what our group really brings into it and what Axolotl brings into it is more on the material side and doing cell biology and actually making tissue constructs that have relevant biology to doctors and other groups trying to study this. So the fact that we can get some of these things like expression of tyrosine hydroxylase, you get to see some electrical activity. We're making some neural tissues that you can actually use as a tool to study these diseases and cell types in an in vitro setting. And for some of the diseases like Parkinson's and Alzheimer's, it just makes quantities of these tissues and human versions of these tissues accessible to researchers. So I'll just show you really quick in case you guys were curious. This is what it looks like when we print with the Aspect.
This is an example of how we have our BSC and printer set up before printing. Unscrew the lid to the printhead and remove it from the container using tweezers.
See the printhead there? And yes, we do print inside of a BSL-2 hood.
Align the 6 holes on the back of the printhead with the 6 posts on the printer. Insert the 4 tubes hanging above the printhead into the 4 holes at the top of the printhead. Ensure that the tubes are inserted in the same order from left to right that they come out of the printer. Push firmly so that the tube is at the back of the hole, but not hard enough to cause damage to the printhead.
Yeah, the printheads are made from silicone. And then because the high-end nature of the system, it's very gentle on the cells.
Open the tubing package and remove one end of the material tubing, ensuring that the tip stays sterile. Loosen the screw in the hole on the printing lid on the Nalgene bottle containing crosslinker. Insert the end of the tubing through the screw until it reaches the bottom of the bottle. Tighten the screw. Take the other end of the tubing out and insert the tubing into the printhead in the crosslinker specific hole. Repeat this step for the buffer bottle and the BioInk conical. Note that the BioInk conical requires longer tubing to reach on top of the printer. Insert the pressure tubing into the printing lid of the Nalgene bottle containing the crosslinker. Repeat this step for the Nalgene bottle containing the buffer and the conical containing the BioInk.
Okay, and so that's sort of how we print with the Aspect system. As you can see, it's got a lot of different moving parts. there. And then the other system that we use is the CellInk, which is a little bit more straightforward. So this is what the BioX looks like.
Preparing the BioX with the BioInk.
And this uses more of the syringe-based systems
First,
for
insert the
extrusion.
female-to-female lured lock adapter to the 3 milliliter pneumatic cell ink syringe. Insert a sterile spatula into the syringe to push the white stopper into the syringe. Take off the parafilm from the end of the syringe containing the BioInk. Connect the other side of the lure adapter to the syringe containing the BioInk. Insert the sterile spatula into the 3 milliliter pneumatic cell ink syringe. Push the BioInk into the other syringe by applying pressure. Unscrew the lure lock adapter from the 3 milliliter pneumatic cell ink syringe. Screw on the syringe tip.
Yes, this is kind of more the traditional using syringe methods. And you can also play with
Place the syringe in the
needle.
BioX printhead compartment.
Yeah, in this case, I think we're using ultrasound gel that's been labeled with food coloring.
Setting up the print model.
So it makes it easier to visualize things.
Select BioPrint. Choose the 3D model that you would like to print. You can preview the model on the right screen. Select Surface and choose a 6-well plate or 12-well plate. Then, select Preview, click on Tool 1, and choose Pneumatic 3 milliliters. Select Layers. And choose the infill percentage.
And again, infill percentage will affect the quality of your
choose
structure
the
being
infill
printed.
pattern.
Same with the pattern. I can also affect the speed of printing too.
Click Print. Printing. Place the desired plate in the print stage. Select Calibrate, followed by manual calibration, and press Start. The printer will now move to the sides and forward and back. Once it stops using the errors, calibrate the printer by positioning the needle as close to the center of the well. The needle should be close to the bottom of the plate, but not touching the plate. Press OK. Then, select the tools and adjust the pressure and print speed. Once this is set, press Start and printing will begin.
You should be getting some movement now, as I think it's starting to extrude some of the gel. We also sometimes use phenol red in our inks just because it makes it easier to see if there's any clogs in the process. So yeah, there you can see it's going back and forth now. And so that's sort of the two different systems we work with. And you can see with some of the syringe-based systems like the BioX, it's a little bit less setup. But as I mentioned, the nice part about the Aspect is you do get really nice cell viability when you work with it, even though it's a bit more work to keep things set up and to keep things sterile just due to the number of parts there are. Okay, and then as I mentioned, one of our recent papers that just came out is we actually looked at using these patient-derived pluripotent stem cells, and we use this as a model for Alzheimer's disease. And in this case, we used one of our smart BioInks. So in this case, we added to our tissue print ink drug-releasing microspheres in this case to print, and these microspheres help promote the maturation of our tissues. And this was just something that was really excited because you can see here in our Alzheimer's groups that had both our control and the one that had microspheres, we actually saw the presence of tau, which is one of the indicators of Alzheimer's disease, as we saw here. And so I think this was on day 30, I believe. And so being able to see some of these phenotypes is something we're really excited about. And then if we can reverse some of these phenotypes, we can use these tissues as a tool for drug screening. And so this was really nice work done by Claire Benwood, along with John and some of the other members of our group. And Claire's actually working at AbCellera over in Vancouver now. And one of the final projects I'll just touch on briefly to show sort of the wide scope of things you can do with 3D printing is, so we collaborate with Dr. Ryan Flanagan's group over at the Vancouver Prostate Center, and he's a medical doctor that runs an infertility clinic. So he essentially sees a bunch of male patients who are unable to produce sperm. And so in this case, we were looking at some of these patients have structural defects, which is why they can't produce sperm. And so the idea was to actually reprint the tubular structure you would find in a healthy male to see if this would enhance sperm production compared to some of the other techniques for making sperm-derived organoids. So in this case, we actually took testicular stem cells. So these were biopsies. So this is a patient's own stem cells. And then we actually did a comparison between what would happen if you 3D printed them versus making a testicular organoid from these same cells. And so this work was led by Megan Robinson, who previously had worked for me and now is now a senior research associate in Ryan Flanagan's group. And so she was really interested in trying to apply some of these 3D printing techniques to help treat infertility. Again, they used in this case the Aspect printer, but instead of using the print heads that I previously mentioned, she actually used one called the Centra that prints coaxial structures. So essentially you get a core shell features you can see here with the red and the blue. And so in this case, you're using the core shell features to actually replicate what a testicular tubule would look like. And so Megan got really nice structures. So she was printing these long testicular tubules from the drawing. And the cells really, really like the bioink, as you can see here with high levels of viability. And this paper actually came out, I believe, in 2002. And if you want, just email me and I can send you a copy of it. Because it was in FNS Science, so Fertility and Sterility Science, the journal. Interestingly, she went and did some gene expression here. And so she found actually compared to the organoids, because we were actually printing structures more similar to those found in vivo and what you would find in healthy men that could generate sperm, we saw increased levels of these genes associated with spermatogenesis. As well as increases in some of the gene expression for cell division to produce the sperm as well. And so this was quite exciting to see, because it was suggesting that the structure does play a key role in enhancing the function of these tissues. And it suggests we were able to get some immature sperm formation inside of these structures, which these were, again, cultured for only 12 days, which compared to some of our neural work is a lot shorter than what you would anticipate. But it was good in a sense because it suggested we were on the right path. And if we could actually fully make mature sperm from these patient-derived stem cells, because we haven't modified them anyway, we would actually be able to Ryan would be able to use the sperm for in vitro fertilization for his patients. So this was really exciting. Megan did some lovely staining here. So again, you can see the coaxial structures. So we did get a core shell tubule here compared to sort of the little circular organoids that we got, and they all expressed relevant markers, although you saw a lot more of these markers in our 3D printed testicular tubule models compared to the organoids. So this is really exciting because this is really, again, along with some of the mesenchymal stuff, ways of actually making personalized tissue models from a patient's own cells and translating them back into applications that have clinically relevant outcomes. If you want to learn more, I know a lot of people have BioXs. In addition to our bioprotocol paper, we also did a STAR protocol paper, where in this case, it was just using our basic tissue print and just HIPSC-derived neural tissues. But the other thing that's good in here is if you want to start bioprinting, it has a recipe for making an alginate gelatin bioink that's relatively affordable. So I would suggest playing with that first and seeing that you can get prints and get your structures reliably, and then moving on to something like our axolotl tissue print product for printing when you actually print with cells, because it does cost a lot of money, not just our bioink, but also when you're printing millions and millions of cells, you want to make sure that your tissues actually survive. For all the grad students in the audience, I do know that's a lot of work to culture and get millions and millions of cells while avoiding contamination and things like that. So you want to make sure that if you're going to print, that you can actually achieve what you would like to in the structures as well. Yeah, as I mentioned, I am the CEO and co-founder of Axolotl Biosciences, along with my co-founder Laura De La Vega, who was co-author on some of the work I've shown, and Melina also splits her time between my lab and Axolotl. So if you're interested in learning more, Anna, our biomedical engineer, she does an amazing job on all of our social media, along with Tom, who's one of our new interns from Portugal. And thanks again to BioProtocol for letting me talk about our work today. I'm always happy to share. I am on Twitter, which I guess is called X these days, but obviously grateful to my whole lab for all the work they've done for all these projects, as well as all of our funding sources, including MITAC, CIHR, Michael Smith, Pacific Parkinson's Disease, and Innovate BC. And with that, I'll stop sharing and we can start to look at some of the questions that you guys have.
Stephanie, if it's okay, I'll just read some of the questions to you from the Q&A section.
Yep, sounds good.
So the first one was, can the bioceramics be the printable bioink?
Yeah, people do print bioceramics. I don't know, I think you could probably do it on the BioX. I think on the Aspect it might be a little difficult given the soft nature of the print heads, but people do print a lot of bioceramics, especially Formlabs has, I think, the Formlabs 3B printer, which a lot of dentists use for printing ceramic work.
And sort of tied into that, what are smart bioinks?
Oh, yeah. So in our case, we like to refer to our smart bioinks as those that have additional functionality. So we have drug-releasing microspheres that help improve the function of the cells seated inside. Other types of smart bioinks include those that change their properties if you hit them with light. I know Cold to Forest has made some of these. So smart bioinks kind of go along with the idea of 4D bioprinting, where the bioinks change their properties over time or potentially remodel in response to the behavior of the cells seated inside. But for us, it's because we have these extended drug-releasing particles in our inks, and that enables better differentiation and function of our cells.
And how do you effectively avoid contamination and decay of the bioink in the system on a daily basis?
Oh man, we do a lot of things. So we do work in a hood is one. We are very, very careful to try and keep things sterile. We do, I believe, use cell lockers where when we transport things between rooms to try and avoid contamination. We wear masks when we print and things like that, just because you're often working right at that level height where you will tend to be getting your hands in and out and breathing. So we take a lot of different steps. I think we spray everything down with ethanol, and we do deep decontaminations of all of our hoods probably every semester.
And I'll add to the, if that's okay, to the decay of the bioink, we really work with how we do media changes so that we're not moving around the construct a bit too much to degrade it or anything like that. So there's technique when it comes to doing media changes as well. What is the minimum size of tissue that can be made using this? So the size, I guess.
Yeah, you might have a better answer to that one. I mean, we're normally printing things that are like 1 centimeter in diameter and like 0.5 centimeter high. I don't know if we go much lower than that, but we usually want to look at like a whole tissue thing. I know other groups have been more into printing like spots of like smaller clusters of cells. I think normally we're like around 100 microns for our filaments. I don't know if they're thicker on the BioX or not. I think it probably depends on what gauge needle we're using on it.
And a really good question too about viability is, how can we compensate for the lower cell viability that we see in bioprinting in comparison to other methods?
Yeah, well, as I said, with the like Aspect system, it's pretty gentle comparatively. Some of the other things are starting out with higher cell numbers to account for some of the cell death that you see. We've also done some printing into baths, which I think can in some cases help viability. Just because it's giving them like a softer place to land on. And again, as I said, like some of the higher, like the Aspect system and the bioassembly bot tend to be gentler on cells because you have better control over flow rates and pressures. I think that's where you see a lot of the cell death is just them being exposed to either shear stress or pressures that they don't really normally see. And for stem cells that are very sensitive, then that's always a big trigger for them. Like as the different pressures and things like that.
I'll read this next one. I am currently working on collateral cancer in animal models here in Nigeria. Can BioX and bioprinting be used in the diagnosis of cancer cells? Can they be used in cancer research, I guess?
Yeah, I mean, we haven't done much on the diagnosis, but Milena's working on, we actually have patient-derived cancer samples that we're printing into our models. We had a really nice paper come out in 2020 in biochemical and biophysical research where Imka, who was one of our star students, came and we printed cancer glioblastoma alongside healthy human tissue and found a drug that would just selectively kill the glioblastoma. We haven't done this yet, but I think it's really powerful that if you're using it for cancer research, you could potentially spike in a whole set of human immune cells into your model for studying it. And so I think that there's tons of applications for cancer. For diagnosis, I'm not as much sure, but you could actually, if you do biopsies, you could do sort of like what we did with the sperm stem cells, because if you do have a cancer cell, and I think Milena, she's worked with all of our cells, would know this, the cancer cells in our ink are so happy and expand so rapidly to form tumors that I think you could actually biopsy and put it into the ink, and then probably you would be able to observe tumor formation. And then also like one of our things would be to then use it as a tool for drug screening, where if you have your, cancer cells from your patient, then you can see if they're actually going to respond to the drug or not.
I will say working with cancer cells is amazing in comparison to stem cells. They're a bit more finicky, to say the least. How do you monitor stem cell quality for suitability for differentiation and bioink preparation?
Yeah, so we do a lot of characterization on our starting populations. We use standardized protocols when we're making our neural stem cells and do a lot of selection and then stain for things like Nestin and SSCA4 and things like that to ensure that we have high quality cells to start with. As I said, some of them, like with our Alzheimer's things, we like to use lines that have already been either published or our collaborators have used that we then know will actually differentiate efficiently into neural cells. As well. And I know that with our collaborators at UBC, I know Francis Lynn's group, who runs the reprogramming core, they've actually been looking into this a lot, is how to consistently make high quality HIPSCs that will get expanded. We've recently started, or we are ordering some of the new Alzheimer's cell lines from Jackson Laboratories, and we also are trying out the new stem cell technologies neural progenitor line as well.
Can you bioprint something similar to a brain organoid? What are the advantages and limitations of using your system for these purposes if doable?
Yeah, so yeah, we've printed things that are similar to brain tissue, definitely for the Alzheimer's work. And then also Ruchi had a Frontiers paper, I think also in 2020 using one of our smart bioinks. So I think some of the advantages are we tend to get more reproducible structures than if you're just making brain organoids, which can tend to be a little hit or miss. And I know that there's a lot of really good efforts out there to make these protocols more reproducible. Our current limitations are size because we don't have vasculature in them yet. And so we aren't mimicking anything like the blood-brain barrier. I know sometimes with brain organoids, you can get some really interesting and complex structures depending on what you're doing. In theory, we could do those, although we haven't done them yet. Our system does have all the major cell types. We do have neurons, oligodendrocytes, and astrocytes. So I think that they're complementary technologies. And we've talked about doing that where we'd print something like resemble hippocampus and then put mature cells around it, because that's definitely doable. The other thing that I mentioned where it kind of also can be complementary is if you're trying to do assembloids where you have more than one region where people make organoids of different brain regions and attach them together, you can use bioprinting to actually put these in the right positions.
I'll move over to the chat. Actually, I just noticed that there's a few questions there. They asked, perhaps I missed it, but have you tried 3D printing cardiac tissue?
Yes, we're currently working on that, and I know Milena's done a lot of work on that as well. We've been moving into that area with Glenn Tibbetts and Zach Laskman, who are at SFU and UBC, and we're pretty excited to work on these collaborations with them. Ruchi actually did do a whole MyTax project on cardiac tissue as well with them. There's a lot of similarities between cardiac and neural, just in terms of needing it to mature, needing to get a whole bunch of different factors right. Being able to maintain phenotype and things like that, but we're pretty excited with where it can go.
And what are the limitations of bioprinting so that one can think of potential solutions?
Yeah, currently the size we can print. So if you can think of ways to add in vasculature is always going to be a challenge, especially because in your body vasculature is supported by fluid flow. I think that's one of them. Resolution, it's not as important for our current applications, but I do know if you want to print something like a lung, you're going to need to be able to print structures smaller than what we currently can do. And so I think that that's also really important. And then I think there's ways to go with just even being able to predict, will this shape print and what it will look like. I think that there's stuff on that end as well. I think there's like a lot of bioprinter technology out there, so I don't know if we'd like more bioprinters, but it will be interesting to see what becomes dominant. Interestingly, a lot of the companies that have focused on the high-end bioprinters seem to be doing not as well as those that have more reasonable models, even though the ones that are on the high end are more likely to actually produce tissues that can actually go into people. So that's something that I thought's been interesting to see.
And if it's okay, I'll also add that it's a bit challenging to run analysis on 3D bioprinted structures in comparison to like a 2D culture. So that's also.
Yeah, and I mean, as I not in a different talk I had, you're really going back to a lot of stuff is like we're actually analyzing these like you'd analyze, you know, tissue samples from an animal because they are like that level of density. So actual like sectioning and staining and that kind of thing.
Have you tried bioprinting with cell lines instead of stem cells, and what are the reasons behind this?
We actually have, and yeah, if you're just starting out, I would actually recommend printing with like more stable cell lines because they're a lot easier to work with than our neural stem cells. But my group is always neural stem cell focused, so that's what we started with. We have about 2 papers worth of really nice skin data, which hopefully we'll be getting out this fall. And the fibroblasts really like it. We've also, I guess we're printing some mushroom stuff this summer. So we printed a bunch of different things there, but yeah, like the skin cells definitely were much easier to print than compared to some of the stem cell type models as well. And as I said, definitely things I'd recommend everyone else try as well.
And if you do want to go stem cell route, mesenchymal stem cells are very hardy cells, so they're a bit easier to work with than HIPSCs or pluripotent. Someone asked, some bioprinters have microvalves such as Reg and View. I have had a very low cell viability with it. Do you think it can be because of the microvalve?
Possibly, because if they're getting exposed to a lot of stress, then that's where you'll see some cell death. So it might be worth trying to figure out like what pressures or what like shear stresses the cells are experiencing due to the microvalve. I don't have too much experience with the REGENHU systems, but I do know they're kind of like the cell or the BioXs in certain ways. So I think that might be worth looking into, is just to see like what kind of stress they're actually being put under. And if there's any way to alleviate that. And if not, then it might be worth looking into some of the other bioink formulations that might make it the process more gentle on the cells.
And a really good question too is, have you faced nutrient transport throughout the printed construct? So how to get nutrients into the construct?
Not yet, because we're still printing relatively small. But I think if we were to go bigger, then that's where you'd start to see the limitations, and that's where you'd want to have some sort of vasculature, whether it's like you know, even just like hollow tubes that you print your tissues onto or something like that. But yeah, we're too small to have that sort of nutrition loaded thing. I know that previously when I worked a lot with aggregate culture, that was something you had to worry about. When the aggregates got too big, they'd get necrotic cores.
And can the same bioink be used to print both plant and animal tissues?
Probably. I know we're just really starting out on this, but we've actually, I think in theory, like our fibrin ink that we really optimize tissue print for neural stuff works amazingly well for a lot of different tissues. But I think with our mushroom stuff, we actually went to a much cheaper thing because the mushroom cells are much hardier. And so I would probably recommend if you're doing plant cells to start with some of the cheaper materials, because I think you would be able to print them a lot more easily than you would say our very delicate, finicky neural stem cells.
And what are the benefits of working with an extrusion-based system like the BioX versus a light-based DLP system?
Yeah, one of the things I think is you get better cell viability without, you know, with some of the light-based stuff, some of the photo initiators can be kind of toxic. And so that was one of the reasons we we wanted to work with that system. Also, some of the photoinitiators aren't really the best for the stem cells. You do get better resolution. It'll be interesting to see, because I do know CellLink has their new version of the LumenX out, and then also there's the NanoScribe. So it'll be interesting to see how they've adjusted to that. And I do know chemistries have evolved. But for us, yeah, with extrusion, it's just been a good way to get really high levels of cell viability and function for our printed neural tissues.
And another question is, how do you confirm the stability of the tissues once printed?
Yeah, so we, I mean, obviously we've worked with our ink for a while, but we've been able to culture them for up to 60 days and they retain their structure. And so that's one of the ways you can track them, is like, if the structure is degrading, is it maintaining its shape, and measuring those properties over time. And so that's kind of how we can assess stability. I know we've also done like mass loss studies and things like that as well, in some of Ruchi's work.
Rheology, all of that.
Rheology, yep.
Solid cancers are spatially heterogeneous disease. How can you address this aspect in printing cancers?
Yeah, I mean, I think that's where it goes towards personalized medicine, and actually, growing each tumor and seeing how it behaves kind of in our 3D system. And honestly, I think it's not just cancer. I think a lot with our Alzheimer's partners and our Alzheimer's work, you see that a lot, that like what we think of as Alzheimer's really has like a number of different causes. And so again, like it really, like being able to work with a number of different patient cell lines, whether it's cancer, whether it's Alzheimer's, it's really going to help give us a picture of like, I think there's going to be the same as with cancers. Like there's going to be certain types of Alzheimer's that will respond to certain type of treatment, other types that won't. And so I think that that's really one of the powerful things you can do with 3D bioprinting, especially with cancers, like looking at these things like at individual tumor level, and then trying to, correlate, like, is this mutation, like, how do we treat this kind of mutation, and that kind of thing.
And which bioprinter do you think would work best for bioprinting two different bioinks, like polymer and hydrogel?
Yeah, it probably really depend. Like I know both the Regenhues and I think the X6s, you can have multiple print heads. So then you could switch between like even extrusion printing and like extrusion plastic printing or something like that, and then printing over like a hydrogel type bioink with them. The Aspect one does allow you to print like multiple cells and would be able to allow you to print multiple hydrogels, but they don't currently sell those printers. But I think something like those, and then I think also the BioAssembly bot also has like multiple print heads and inputs, so you could use that to print multiple different ones side by side. But I would suggest always like going and potentially trying these out first before going ahead and buying such a system.
And a bit more of a techie question, I guess, is how do you mix the cells in bioink?
Don't we just add it to the ink directly and then try and use it immediately?
Yeah, so usually it's a timing thing. We usually have the cells in culture, and then we passage the cells the day that we're printing. So you sort of have to have a very strict schedule in terms of knowing when your cells are going to be ready. And then once we passage them, we simply just incorporate the fibrin. We mix them with the fibrin and incorporate it into the bioink. It's actually relatively easy, easier than expected, I'd say. Have you done work regarding smart materials or thermoresponsive materials?
Yeah, so as we said, we have our drug releasing microspheres that we've incorporated in there. We haven't done as much with the thermoresponsive materials. I mean, I guess in some cases you could consider the bath kind of thermoresponsive, but I do think it's an interesting area to be explored. And I also think there's been, like, I know Cold DeForest Group has done some really cool things with some of their 4D bioinks that they've made from just a more pure chemistry science background. So it's definitely of interest. I also noticed, we have one person with their hand up. I don't know if we want to actually call on people with their hands.
Sorry, I can't see that for some reason. Renee, would you
want me to allow it? So I think it's for us. If you're okay, I'm going to click allow to talk. So hopefully. Okay, I think we're good. You should be able to ask your question. Nope. Guess not. Okay.
go to another question in the Q&A is scalability and affordability a huge problem when looking at the aspect of incorporating this technology for medical use?
Yeah, I mean, I think the biggest thing, and for everybody in the audience, this is kind of one of the things I always put out there. Lots of cells. We need to find a good way to scale up how we expand our neural progenitors without just having students or employees do it manually. So the cell is one thing. We've gotten better at scaling up our BioInk production since we've moved to having a company. But I think to go to the point of making liters and liters of BioInk would have to move to a more process-based method, which would be chemical engineering challenges. But I don't think it's anything that couldn't be overcome. One of the things that's come with COVID and other things, being able to source materials has become a little bit difficult. I know we've been chasing different types of collagen providers and things like that, and then also Matrigel has been in short supply. So I think that there's some of the issues on that end, like how do we actually get our starting materials, and we've had to switch some suppliers in the past year to deal with that.
What are the BioInks immunogenic? Can we test immunotherapy in printed 3D cancers?
Yeah, so our BioInks are xeno-free, so they don't have any animal products. So in theory, you could make a GMP version and put it into humans, which would be really exciting. And one of our beta testers at Axolotl is very interested in doing that. And yes, actually being able to test immunotherapy for 3D cancers is something that I think is a really, really exciting potential application for our systems. As I mentioned in one of our papers, we actually did do human cancer alongside healthy human astrocytes, but for all your CAR-Ts, this is where you could actually have human on human cell therapies. Some of you may know the way currently people test cancer therapies is you take a human tumor and you put it in a mouse with no immune system. And so it's a very, very artificial system, but our systems and 3D printing allows you to have a system where you could actually spike in your different immune cells. And we've been starting to do a little bit of this on the neural side, looking at microglia and incorporating them in. Obviously, the immune system plays a big role in how you respond to all sorts of different types of treatment. But I think that that's something that's very, very exciting for the cancer systems, is that you can have the human immune cells, or as you'd probably be alluding to, an engineered CAR-T, and you could see, is your CAR-T going to actually kill the tumor in a tissue?
And I guess I think I saw a question earlier and I can't find it right now, but in terms of co-cultures and sort of the how do you do co-cultures and which media do you choose? Or if you have I think they were mentioning endothelial cells and something like smooth muscle cells or something like that. And which media would you use for co-cultures?
Yeah, I know for our skin work where we did do co-cultures, it was a lot of trial and error to figure out what would work best. For our neural cells, we usually go with what the pickiest cell type is and then use that specialized media for that. So if you're going really different, then yeah, I would say you're probably going to be looking at a little bit of a media optimization exercise to get that to work like we had to do for our skin cells.
Have these bioprinted tissues been used for treatments already, or are they still in trials?
Yeah, as I said, the one that's, I guess, technically a treatment, but I think still a clinical trial has been the 3D printed ear one, which is again with 3D biotherapeutics. I don't think other tissues have gone clinical yet, just because I know for some of the diabetes work, when you're looking at putting that many cells into a person, it's going to be looked at, the bioprinted tissues looked at both as a cell therapy and a device. So I think there's still a little bit of ways to go, but the fact that they've already got sort of one of these products approved and in clinical testing is really positive. I think you're going to be more likely to see it for things like the cosmetic version of the ear. I honestly think skin bioprinting for clinical applications will be quite easy, just because it'd be easy to print on a tissue in that sense, and skin isn't super critical, if you're dealing with something like the heart. For neural, I don't actually think they'll ever probably go therapeutically. I think you'll probably just be doing cell injections. And I do know there's been some really exciting work with the cell therapies that have been really positive for Parkinson's. But yeah, just getting into either spinal cord injury or the brain, yeah, you aren't really usually going to be replacing a whole structure. Things way down the line, like a heart, you're okay, that makes a lot of sense. You could scan the patient's heart and then rebuild it from scratch. The same with lungs. Those seem to be if we actually get all of our cell biology and things worked out, those would be great tissues to explore.
What about bioprinting for cultivated meat?
Yeah, no, there's a lot of interest out there in that. I think Ali Khan Husseini's group has an O-Meat version. Again, the big issues are getting the cost of culturing cells down. And I do know Ollie's group, and then there's another group out of the Netherlands that have worked really hard at, finding things to make serum cheaper, to make all the different components you need when you're culturing human cells cheaper, because that's one of the big barriers, is like the high cost of reagents. And obviously Melinda knows, and probably a lot of people on the call know, doing stem cell work is not cheap at all. And so like being able to drive those costs down, because your main thing is going to be getting enough cells to print physiologically relevant issues. The other challenge is for a lot of those tissues, and this is not just beef and chicken, but I know people are also looking into bioprinting seafood. It's just that the biology is relatively understudied compared to things like human-induced pluripotent stem cells. We often get like calls to consult on them, and it's always like, well, we don't actually know this about like chicken embryonic stem cell biology. And so, it's just, I think, working with some of the systems that are less common and dealing with those.
And I think I'll do one more question since we're getting close to 10. Does 3D printing make errors such as mutations?
Not directly. I could see like issues where like, the stress and like shear stress could potentially induce a mutation just through the stresses of the process. And with any cell line, it's kind of important that you always, karyotype at some point just to make sure you're not carrying through as the cells divide. But I just don't think, like, not necessarily directly, but yeah, potentially as a result of stress and things like that. You could definitely see those kinds of things.
And I would say also if you're using like UV,
Oh yeah,
things like that.
All right. Well, excellent. Let's end here. Thank you so much again for a very interesting talk, Stephanie and Milena, for great moderation. And thank you everyone for a lively discussion. We had almost 50 questions, which is amazing. If you have more questions, please feel free to visit our webinar website, where you can still post some, or also participate in a discussion if you are an expert in this space. And as I mentioned earlier, this webinar is also being recorded and will be posted on the website later. And lastly, I would like to announce our next webinar, Mic Drop, a platform for large-scale in vivo CRISPR screens, by Saba Pervis, on September 5th. And with that, thank you again, everyone, for coming, and I hope to see you next time.
Yeah, thanks, everybody. That was awesome.
Thank you.
Recording.